So we're going to start that now. So welcome to the 2023 Soil Health Webinar Series. We are a little behind today due to technical difficulties, but you know, it's January and we'll work through that. Today, we will be talking about precipitation management and the effects on soil health with Peter Tomlinson from K-State. We appreciate him being here. A few housekeeping things that we have for you today. There will be a soil and water management CEA credit available. This will be offered at the end of this session. This is only available on the live videos, not on the recorded sessions. These recorded sessions will be available at agcrops.osu.edu for viewing at a later date. Please feel free to submit questions at any time using the Q&A chat feature at the bottom of your screen. You can also email questions to myself, Jamie Hampton, at hampton.297@osu.edu. We will use the chat feature and poll questions to get feedback from you today. So our speaker today is Dr. Peter Tomlinson. He is from Kansas State University, and our moderators today are Sarah Noggle, A&R educator from Paulding County, and Gigi Neal, A&R educator from Claremont County, and myself, Jamie Hampton, A&R educator from Auglaize County. So at this point, I am going to stop share on my screen and turn this over to Peter.
All right. Give me 1 second to get into presenter mode here. All right, are you all seeing my slides yet?
No, we are not yet.
Right.
There we go. We've got a nice view of your whole screen, but the presentation is not in presenter mode yet.
Okay. One. Come on, Peter, cooperate. There we go. That working now?
Yep, looks pretty good.
Great. Well, good morning, everybody, and I do apologize for the technical difficulties. My Kansas State University email and the Ohio State University Zoom system were not in agreement this morning. So thank you for your patience, and thank you to the committee for the invitation to have the opportunity to spend the morning with you all and share some of the work that we have going on in Kansas. So as we think about soil health properties, how to assess them, Kansas presents, I feel, a unique opportunity in that we've got quite a precipitation gradient across the state. And so as we're thinking about these soil health properties, we can really think about it in terms of a couple different directions. We can think about it in terms of, the three components of soil: the physical properties, the chemical, and the biological properties. But we can also think about it in terms, and that really gets to the point of the soil as an ecosystem. But we can also think about it in terms of, what are the intrinsic properties of a given soil, mineralogy, texture. These are properties that are not easily altered by our management practices, or we can think about more dynamic properties. And so as we think about dynamic properties from a chemical standpoint, soil pH, what is the soil's fertility capacity, organic matter, soil biological properties, and these are properties that are assumed to be responsive to management. And so, when the Soil Health Institute started their large nationwide project, they looked at properties in a number of different ways. And what I've got here is a table that they put out in 2018 of what they felt were tier 1 soil health indicators and the associated methods. And these were indicators that were widely used in soil testing laboratories, had a high degree of confidence around how to interpret the results. And so we see things like pH and EC, extractable phosphorus, total nitrogen, soil organic carbon, coming out the chemical indicators, physical, soil texture, aggregate stability. And then in the biological pool, I've always found this interesting, the tier 1 indicators that they included were crop yield. And I think that is frequently, an indicator that we don't necessarily think about much in soil health, but really, we think about that growing crop as our key indicator of what's happening. I think it can be an important tool. Short-term carbon mineralization, nitrogen mineralization, these were all the tier 1 indicators. And then, they had a tier 2 table, which was indicators that had been used in a lot of research laboratories. And we'll get into that a bit more. But around the same time, the USDA NRCS issued their soil health technical note, recommended soil health indicators and associated laboratory procedures. Around this same time was I just saw the note. Let me see if I can adjust that share. Is that any better on how it's coming through?
No, Peter, I think it's a double screen.
Okay, hopefully that will be better now.
That's correct.
Okay, so as NRCS put out this technical note, they lots of research questions came out, and so it's with my background in soil ecology and colleagues here at Kansas State, looking at these soil health indicators has been something we've been working on across a range of projects. And so, I've just got the precipitation gradient for the state of Kansas, and you can see on the western side of the state, over here, we're in that the light yellow, which we don't have any. I won't be sharing any research sites. We're 13 to 18. In this kind of lightish green, 18 to 23 inches of annual precipitation. And then as we move east across the state, our precipitation increases, and I'll be sharing some studies from the central part, and the upper 20, low 30 inches of precipitation. Then on the eastern side, where we're in that 36 to about 40, 45 inches of precipitation. And so the first study I want to share with you is one that was funded by the USDA NRCS Kellogg Soil Survey. It's a dynamic soil properties study, and it was located in 3 western counties out here in western Kansas. And what we looked at here was the Keith Soil Series. It's an important soil series in western Kansas. It's considered one of the benchmark soils in Kansas. And, you know, it's a loess wind-derived soil, but 80% of the acres are in cultivated cropland. We've got about 20% that are still in intact native range. This soil sits over the Ogallala Aquifer, so irrigation overcomes the moisture limitations and you know, really makes this an exceptionally productive soil. And so, you know, we do also have a fair bit of dryland wheat and grain sorghum growing on the Keith series. And no-till has become prevalent in moisture conservation and the management. And so, you know, these are the locations of the sites in those 3 counties in western Kansas. And this just starts to show the extent of the Keith Soil Series in the area where we did our sampling. And so we wanted to look at 3 levels of management. So we wanted to look at, you know, what if the soil was still under native prairie? And so we identified native prairie remnants in proximity to other management. And so we've been able to set up kind of a nice continuum going from native prairie to long-term no-till with either, you know, cover crops or diverse crop rotations, bringing peas back into the rotation, and then very traditional conventional tillage as the other end member along that continuum. And so we sampled these soils down to a meter. What I'm going to focus on is the results from the 0 to 5 centimeter samples that were collected. We worked with our local NRCS and did producer interviews and soil health assessments. We won't get into that today. But our sampling plan here was within a field. We wanted to stay in the same landscape position, and then, you know, collect, you know, three discrete samples within a cluster. And so you can kind of see that here at the top of the screen is our cultivate or our long-term no-till in the north cluster, and then down here is a native pasture remnant that we were able to sample. And so what are some things that we found? So my colleague Deanne Presley did the work on the aggregate stabilities that I'm sharing here, but we looked at a number of different methods. Looked at, you know, the traditional ARS method, multiple sieves, mechanical shaking, and then the Kellogg Soil Survey has a simplified single sieve method. And we begin to see there's some differences. You know, the ARS method, you know, separated out conventional from the soil health managed soils, and the statistics group the soil health and the rangeland soils as being more similar than the conventional. However, if you look at the Kellogg Soil Survey single sieve method, we see that, that's not what we're how things are grouping, and the conventional and soil health group together, and then the rangeland is separate. And so, Deanne feels that, that multi-sieve method is still very much the superior, or the gold standard method for doing aggregates. Where my group took over was, as we started to look at chemical and the biological properties. And so from the standpoint of soil organic carbon, interesting result here. The rangeland, which we would have expected to have the greatest soil organic carbon in the near surface, at almost 3%. But we actually, measured, conventional as the intermediate in this system, followed by the soil health systems across the 3 sampling areas. And I think part of this is that, as we think about, the moisture limitations in the region, the development of soil organic carbon can be slow. We do have some accelerated biological activity in the soil health system. They could be consuming some of that organic matter, whereas in the conventional system, we observed we had burial of crop residue and slower decomposition. And so in terms of some of the biological properties, we looked at respiration and POC. Respiration in rangeland separated out from the 2 managed systems, cropland systems. But for POC, we found, again, rangeland had the greatest POC, but then the conventional, followed by the soil health system. So that doesn't necessarily agree. You'd think that we'd see potentially a higher respiration rate, given what we were seeing for soil organic carbon being lower in the soil health system. So, what else could be going on? And so from that standpoint, we wanted to look at enzymes. Beta-glucosaminidase and beta-glucosidase are enzymes that are involved in carbon and nitrogen cycling. And again, what we see here, the rangeland separates out from the conventional and the soil health systems. Again, the soil health system, in terms of beta-glucosaminidase, actually had lower enzyme activity compared to the other 2 managements. We did not see that for beta-glucosidase. We also looked at enzymes involved in phosphorus cycling. And again, rangeland separates out, and the conventional and the soil health management systems, typically we're going to see that there's not a lot of difference. Acid phosphatase, and we'll see acid phosphatase come out in some of our other studies too. We see, the conventional system there had it was both similar to the rangeland and the soil health system, kind of an intermediate there. We also did PLFA analysis, and for those that aren't familiar with PLFA, that's phospholipid fatty acid. It is basically we're extracting the in using the cell structure of the microorganisms to be able to look at the diversity of the microbial community. It's a coarse measure, but when we look at some of the other molecular methods out there, it's a cost-effective method that can be employed to start to look at and get an understanding of the microbial community that's at play. Not surprising, rangeland, when we think about this in terms of total weight or biomass size, rangeland had the largest microbial community. The soil health and conventional systems really were not different from each other. And then we can look at subgroups. So arbuscular mycorrhizal fungi or AM fungi, gram-negative bacteria, gram-positive bacteria, the pattern follows. And we've got more work to do here to look at, do we have, really major differences in the microbial community structure? Is it just a matter of the size of the microbial community? But at least here we're fairly consistent in terms of rangeland having the largest both total biomass and then biomass by subgroups. And so rangeland separates out. We have a few differences between the soil health and the conventional system. This was a bit surprising. These soil health systems had been in no-till for 20 years, had had that diverse rotation or cover crops for the last 9 years. But we think that the low moisture is a real driver here, and the high evaporative demand in Western Kansas that really slows down the potential for change. And so let's move east, and we're going to look at a study that was in place for about 15 years in Heston, Kansas. This was work that was done before I joined Kansas State University, but it was kind of a labor of love project of Dr. Mark Clausen, and a number of colleagues worked with Mark to get this work written up at the end of his career as he was retiring. And this was a winter wheat grain sorghum rotation with cover crops. He had summer legume option after wheat. He looked at sun hemp, as well as some alternatives with cowpeas, soybean, and mung beans. And at the end of that 15 years, was able to document some, really, significant, what I'd consider significant changes. But again, we see these changes really in that top 3 inches or top, 7.5 centimeters. Soil organic carbon increased in both the treatments with the late maturing soybean as a cover crop and the sun hemp compared to the no cover crop. And with this, we can also look at it. There was a nitrogen rate component to this study. And as we increased the nitrogen that was added to the system, that also translated into an increase in soil organic carbon. They also did infiltration work, thinking about how water is moving into the soil. Now this is near surface, and I think we always need to be careful when we look at infiltration rates that, that's looking at a very small portion of the field. We need to I don't feel there's a direct, in some of our watershed work, necessarily a direct correlation, but it is an important indicator of how the field is behaving and how the water may be moving. But again, we see that with the sun hemp and the late maturing soybean, some sun hemp increased the cumulative infiltration rate, certainly compared to the control and the late maturing soybean. And another interesting thing is they did some earthworm survey work out there and found that the greatest earthworm counts were in the two cover crop treatments. And so, this wasn't done right after the cover crop. It was done after the grain sorghum harvest. So, that represents that it had been, close to 12 months, probably closer to 14 months since that cover crop had been present. But it does show that we've got, as another, if we think about earthworms as a indicator species, there's certainly something going on here that is attracting the earthworms, likely as the cover crops. And so we'll move further east and towards more moisture. And we're going to look at, as I finish up my presentation, we'll look at two studies, one here in the Manhattan area, and then one a little bit further east at our experiment field by Topeka. So our Kansas River Valley field by Topeka, the station director, Erica D., had started a tillage study. And why tillage and soil health? Well, I think that how we manage our soil is a very personal decision to each landowner. And so in my role as an extension specialist, I want to, have information for all. And so we thought this was a unique opportunity to, look at a no-till versus tillage comparison. And see, how does in an irrigated system, how does tillage affect both soil carbon stocks and dynamic soil properties? So it was a irrigated corn soybean rotation that was started in 2013, and we sampled in 2001. And so in terms of physical indicators, total aggregation, and I'll set this graph up. So our no-till, as we think about a continuum, would be our least intensive system. That fall sub spring vertical till would be the most intensive system. So that's fall subsoiling tillage pass followed by a spring vertical till that's occurring before corn planting and before soybean planting. So basically you're looking at 4 tillage passes in that one. We've got fall vertical till ahead of the corn and the soybeans. So 2 tillage passes. And then we've got a fall subsoil ahead of soybeans and a fall vertical till. Actually, I think that's after corn and then the fall subsoil after soybeans. So 2 tillage passes there. And interestingly, what we see here is the one that stands out in terms of total aggregation is that fall subsoil spring vertical till. So our most intensive, and it makes sense. We've got most intensive tillage and it's standing out. It's got the lower total aggregation. So when we looked at the carbon stocks, though, surprisingly, we did not measure any significant differences in carbon stocks. You can see by these box plots, we've got more variability, but statistically, there's no difference whether we looked at total organic carbon in the top 2 inches, which would be the light gray bars, or the top 3 inches, which would be these hashed bars, or what looks as just black lines here at the bottom, the permanganate oxidizable or POX-C, and I've blown that up over here to the side. And again, we've got some differences in the amount of variability, but we're not, measuring a significant difference in the carbon stocks. So what if we look at enzyme activities. And so the one I've highlighted here is beta-glucosidase. And interestingly, it picks up that most intensive tillage practice decreased the beta-glucosidase activity. And so, from that standpoint, we're not, I didn't share the data, but we've not seen clear differences between our tillage systems in terms of yield results. Carbon stocks did not seem to be affected. But certainly when we think about the soil health standpoint, that management system with the greatest intensity did reduce total aggregation and beta-glucosaminidase activity. And so the final study is our Kansas Agricultural Watershed Field Laboratory. This study was set up in 2014. It is comprised of 18 1.2 acre watersheds, and we've got a number of different activities going on in the site. We're collecting runoff after every precipitation event. My research group has been doing soil health work out here from the beginning. And so, it provides us a unique opportunity. One of our real water quality questions is, around phosphorus management. How does, fertilizer source and placement affect not only water quality, but also soil health? We've got it set up in such a way that we can look at fertilizer placement by cover crop, or with a cover, is there a cover crop interaction? And so, again, colleague Deanne Presley, her group has looked at physical properties, and over the multiple years, we've seen higher mean weight diameter in the cover crop compared to the no cover crop in 3 out of 4 years. And then in terms of what are we seeing in terms of changes in the size of aggregates? This would be the graphs over here on the right side. We've got we had more, I would say, more small aggregates in 2017 than we certainly have now in 2022. And interesting, we didn't, in that smallest size fraction in 2017, no statistical differences, but now in 2022, we've got more of those small aggregates in the no cover crop compared to the cover crop plots. And we've got, similar to what we saw in 17, we've got greater number of large aggregates in the cover crop compared to the no cover crop. So, we certainly are affecting the physical properties by the addition of cover crops. Did not see an interaction with phosphorus management practices. And so, my group has looked at number of different biological indicators. And so, we've done, looked at the microbial biomass. This is based off of phospholipid fatty acid extraction of the soil, and we see greater microbial biomass. This is just snapshot from data in 2018, but in both the spring and the fall, greater microbial biomass where we had cover crops. Not surprising, we saw the greatest increase in the spring. This would have been right after cover crop termination, so we would have had that growing cover crop all winter. But that legacy effect carried through the growing year when we sampled right after harvest. very similar patterns with our carbon cycling enzymes. So both the beta-glucosaminidase and the beta-glucosidase. And we see that, higher enzyme activity with the cover crops in the plots with cover crops compared to the treatments without cover crops. And we looked at the suite of phosphatase or phosphorus cycling enzymes and the same pattern. So, in this work, certainly cover crops are playing, an important role in increasing, some of these dynamic soil health properties. We also looked at the diversity of the microbial community. And this was a real interesting one for me, in that, I thought, okay, we've got system managed, all no-till with cover crops and without cover crops. And I really expected that we would see some shifts in the microbial community. And really, we didn't see a shift in the microbial community. We have larger microbial communities in the cover crops, but the relationship of the community members stayed consistent. And so my current PhD student has had some questions, what are the seasonal dynamics of these soil health indicators? And, are they, how are they changing over time? And so soil respiration, microbial biomass carbon, active carbon, we were not able to pick up a cover crop treatment effect by time interaction, but we did pick up that time was a significant main effect on these indicators. And we can see, a lot of this is driven by precipitation and soil moisture. As we get into the drier part of the year, we're seeing, a decrease. Then if we start to catch some late summer moisture, we're seeing some increases as we get out to harvest. And so, this gets back to the precipitation question that, the rainfall and the precipitation patterns, I think, do play an important role in the what we're able to measure and what we're capturing in terms of soil health indicators. And so the one that we did capture a cover crop by sampling time interaction was for acid phosphatase. And, we consistently saw greater phosphatase activity in the cover crop treatments compared to the no cover crop treatments. We see, with these box plots, we see greater range of variability where we have the cover crop compared to the no cover crop. But, if we're trying to pick what is the right sampling time, we're going to get, at least with this indicator, we're going to generally get the same comparison, if the question is, what's the difference between, a system managed with cover crops and without cover crops. And so, summarizing this work, soil health indicators are positively affected by the addition of cover crops. As I said, microbial community structure was not changed by the addition of cover crops. And, moisture, temperature, all of the environmental factors, do affect what we're able to measure with the soil health indicators. And so I think, as we start to think about, what is the right time and place to do our sampling for soil health indicators, I think the biggest takeaway that I have is we just need to be consistent from year to year and make sure that we're documenting, what are the environmental conditions when those samples are being taken. As we think about the precipitation question that was part of the overall theme here, I think as we head, here in Kansas, as we head west, many of these properties take time and are slow to change. Like soil organic matter. And if you add a high evaporative demand and low precipitation, I think you're going to slow that process down even more. But there are opportunities, I think, certainly with some of the physical properties, like aggregate stability, by the addition of, say, cover crops or conversion to no-till, to see more immediate results of your management. I think it is important that we think about, how do we protect that soil surface? We know that's important, from helping to foster a robust soil ecosystem. But it's also important when we think about precipitation capture and storage, protecting the aggregates at the surface, protecting the large macropores so that we have the ability to infiltrate water and reduce I didn't show this data, but with the watershed project, we've been able to document some important reductions in sediment loss. All of these indicators are dynamic. When we sample, what are the conditions around them, is influenced by precipitation, by temperature, freeze thaw activity plays an important role. But I think we're looking with a long-term mind frame. There are opportunities across all environments to build our soil health. The payoff may not be immediate, but I think in the long run that investment will pay off. And so with that, I thank everybody for their time. Sorry, I rushed through some of these slides, but happy to take any questions.
All right, thank you very much, Peter. I am going to have Sarah go ahead and run our questions. Looks like we have just a handful of questions in our Q&A section. So Sarah, if you want to take over for that.
Sure. First question is, this was kind of at the beginning of the presentation. How long was the soil health fields in their practices? Did the farmer change practices from conventional due to poor soil health?
Yes. So, and I'm going to assume that is in reference to the first study that I shared in Western Kansas. Those the fields that we considered our soil health fields. When we were looking for producers, we looked for producers that had been early adopters of no-till. So all of those fields had been in no-till for at least 20 years, and had in the last 10 years had either started to incorporate cover crops into their crop rotation or going to a very diverse crop rotation that included peas, not your traditional, wheat and sorghum, but we're looking at, in some cases, 4 different crops in their crop rotation.
Okay. Were cover crops included in the rotation at the Kansas River tillage study?
No, cover crops were not included in that study, and that has been one of the as we've been looking at this data, that's been one of the observations that, boy, it would be interesting if we had that cover crop comparison there, but unfortunately we don't. That one had the advantage of irrigation though. And so, I think that, one of the things that we see there is, I think, particularly in terms of the carbon stalks, the irrigation, I think plays a role in why we're not seeing some of those differences in the carbon stalks, because we're able to see consistent yields year in and year out.
All right. Is the difference between the aggregate size in the 2 years attributed to the dry weather pattern?
I'm going to assume that is in reference to the Kansas Agricultural Watershed Project slide set. And no, I don't. I think that aggregates, looking at year to year, you've always got to be careful because weather does factor in there. But, that represents about a 5-year management spread. And so when the Kansas Agricultural Watershed Project was started, that project gave us a unique opportunity. That field had been in full tillage foundation wheat production before our project took it over. And so we've been able to look at how soil health properties develop over time, and and so I think that what we're seeing there in terms of the aggregates is we are seeing the effect of time, not necessarily precipitation, because yes, we've had some dry years in there, but we're looking over, you're looking at about a 5-year time spread between those two samplings.
Okay, the next one, it says, can you talk more about the composition of cover crops in the last study? Are they cover crop monospecies plots, or do they include multi-species and different types of plants? I'm wondering how different types of plants, grasses, legumes, and forbs influence soil health indicators.
Yeah, great question. That study has predominantly, because we had lots of questions around erosion, has predominantly been a small grain winter cover crop. We've had wheat, cereal rye, triticale, over the now close to 7 year, or longer than that, 8 years of the study. But we have tried to diversify that. We have included, brassicas in the mix. I will say, just with the timing of harvest of corn and soybeans, we've got our best chance to get cover crops up after corn harvest. That gives us our greatest fall growth opportunity. But even with that, we've struggled to really get the brassicas to establish very well in the fall. And so the end result has been, it's predominantly been the small grain. But that said, we have another study in the same area, and I've got a student working on that right now, where we've got a diverse cover crop mix, crimson clover, cereal rye, and a cereal rye crimson clover mix. And so we're working on looking at differences in soil health indicators with those different cover crop species. And so I'll have some more information on that to share in the future.
All right. It said, on the recent grad student work, Ari, the timing effect on carbon pools and microbial communities, what are the T0, T1, T2, etc.? Are those hours, days, or months?
Yes. Sorry, I apologize. I was trying to hurry there and should have provided that detail. Those represent, so T0 would be at cover crop termination, and and then each time interval is approximately 20 days. So you're looking at out through T4, so about 80 days into the corn grow, into the corn year, and then that T5 is our fall at harvest sampling.
All right. This one's probably for our team. OSU had a soil health sampling program recently where I participated and received results. This was discontinued, wondering if there was consideration of this being renewed. Also, will there ever be a general summary of those results? On that one, due to Dr. Steve Coleman leaving, that part of that study was stopped, but we can get you some information. If you want to email either Jamie or myself, we'll have Jamie's email there at the end of the presentation, and we can let you know, but we'll find out that answer because I'm not 100% sure. All right, the next one. Do you think the addition of soil biologicals that are offered on the retail market today are likely to be economically beneficial to growers?
Oh, that is a good question. I have not had an opportunity to do much research with biologicals. I get lots of questions about biologicals, but companies have not, when I've been asked, companies have not tended to, or have not to this point been willing to make the research investment in field work that I'd be able to share information in extension programs. When we think about the microbial community and the diversity of the microbial community, we've certainly got we've got a lot of microorganisms in our soil. But we also know that when we look at, inoculums for legumes, that, does it pencil every year? No, but it is really, from a risk management standpoint, is really important in making sure that we're putting that inoculant out every year. So is there potential with soil biologicals? I think there's a possibility, but I think it's an area we need a lot of work, and I don't know what the right way to approach that from a research standpoint is.
And I can add into, through Ohio State, Dr. Jane Fife, through the college, she is actually working in some of that area and looking at some of those research standards. She will be speaking at Conservation Tillage Conference in Ada in March, so that might be a session to attend. Let me get you the next question. It says, were there any water infiltration data taken in the different systems?
Yes. So there was, we did with the Dynamic Soil Properties Project in Western Kansas. We did take both single ring and Cornell infiltrometer measurements. I do not remember that data off the top of my head. At the Kansas Agricultural Watershed, we have not taken infiltration measurements per se, but we know the area, and we have multiple rain gauges across that 18-acre site, and we know the runoff. And the interesting thing that we've been able to look at is the hydrograph data one of the interesting findings that we've seen, and this has held true the last several years, is that our infiltration, well, the infiltration is tricky. What we see is that where we've had cover crops, we decrease our peak runoff volume. So if you think about that in terms of, it's how much, what's that maximum amount of water that's coming through the flume. And so we've decreased that peak volume. So, we're slowing, that tells us we're slowing the water down. And then we've increased the duration of the runoff event. So that runoff event is lasting longer. But the interesting thing is that when we look at our yearly summaries, and it a bit depends on what the soil moisture conditions are preceding the runoff event, but generally we see the same amount of runoff off of the systems with and without cover crops. We're just changing how that water is behaving. And so we think that, and I didn't get into that, but from a water quality standpoint, we think that that plays an important role in some of the phosphorus loss patterns that we're seeing. And I'd be happy to share that in another, that's a whole nother hour.
Exactly. Does cover crop termination have an impact on herbicide versus disking?
Okay. So I'm not a weed scientist. I'll put that disclaimer out right up front. But I do work with some great weed scientists here. And we've been working with our ag econ department to do some of the economic analysis. I think cover crops are a good tool and do provide some opportunities to potentially reduce a herbicide pass. And it may not necessarily result in the reduction of a herbicide pass, but when I've heard our weed scientist, Sarah Lancaster, say is our weeds tend to be smaller where we have cover crops. So there's a potential to increase the effectiveness of the herbicide pass because you're not trying necessarily to kill big weeds, but you're killing, you're targeting smaller weeds that are in a more susceptible growth stage.
All right, we'll do one more and then we'll share the CCA credits and then we'll continue with those questions. In the soil health studies I did, there was a difference between long-term conventional tillage versus modified conventional on the percentage of bacteria versus fungi in the system. Is this consistent or just an anomaly due to minimal sample size?
Brian, it's a great question. I'm working on that. I would have expected, I would expect that where you've got, conventional tillage and then you're going to reduced tillage, I would expect that you're going to change the microbial community. We did not see that in the Western Kansas data. And I'm not, that's something I'm drilling down on more. It's certainly, something I've observed in other work that I've done. But I think in terms of the data at the Kansas Agricultural Watershed, those are both no-till systems. The only difference is the cover crops. And so I think that's why, I don't know I think that's, it's supporting a larger community, but we don't have that major physical change driver there. So, it's certainly something that there needs to be more work done on nationwide, but I don't think it's an anomaly. I think it's just, you know, as we get into the microbial community structure and those microbial dynamics, I think it's an area that we just don't have a lot of data to really understand.
All right. Thank you very much. I am going to stop recording.